So, Kerrie, I've just discovered you and I were at the same university, so I'm really excited to find out what somebody actually learned, because I wasn't great at attending stuff. So, Kerrie, Miscanspeed, you're going to talk to us about genetic selection and speed breeding to accelerate the development of resilient Miscanthus varieties. So, looking forward to it. Thank you.
Right, this is Aberystwyth. This was our first field of Miscanthus. Now this is quite an old picture, so we've filled up quite more of the land with Miscanthus, and we're obviously all aiming to do more of that. So just to say, this Miscanspeed project, it's a £2.1 million project, very grateful for, and it's really looking at genomic selection to accelerate the breeding of Miscanthus, which is something that we've been doing for some time at Aberystwyth. And it fits into a scheduled work, including some of the more fundamental and strategic crop research that we do at Aberystwyth, and then also some of these other bigger programmes, more in the sort of demonstration and understanding more about the greenhouse gas removal components of the crop. But today I'm just talking about Miscanspeed. So, as I said, we've been doing Miscanthus breeding for a while in Aberystwyth. We know the drivers. We need really resilient Miscanthus genotypes to produce high yields over 10 to 15 years for future climates, which is obviously a challenge. And so we really need diverse genotypes so that we can have a really resilient biomass supply chain. And the approach that we've taken is Miscanthus is a new crop, it's not domesticated, it's a wild species. So we've collected germplasm. This is the engine of a breeding programme. You make selections, you evaluate the progeny, make selections, send them out for multi-location trialling, and ultimately you end up with some new varieties. And as I say, this is what we've been doing. So we have large collections that we've made from Southeast Asia. We've resulted in the first new biomass varieties available in Miscanthus, and concurrently we've been working on that really important scale-up and seed production and agronomy for Miscanthus. So that's the background and why we're well placed to do this. And you can read a bit more about this in the UK Plant Science Research Strategy if you're interested too. But we've got some challenges that we really need to address as well. So Miscanthus takes 3 years to reach maturity, so that obviously limits your breeding capacity. The phenotype's dependent on the environment, obviously. So again, as Ian was just talking about, you need those multi-location data to really understand how that's going to affect it. One of the challenges we have is that different genotypes flower at different times, so just even if you know which crosses you want to make, physically making them can be quite difficult. And all those different species have different regulation for the different components of their life cycles as well. So there's quite a lot to grapple with. So in Miscanspeed, we've taken the approach that we want to address some of those challenges. And again, we've come to genomic selection as the approach we want to take. And this is where you take a training population, you combine the phenotype information with the genotype information for the plants, as Ian was saying. You combine those to make genomic prediction models. And then instead of doing the central part based on the phenotype of the plant, which you have to wait for 3 years to achieve, you can do that at the seedling stage based on the markers instead. And what I should have said is, importantly, that introduces us to a new challenge, because usually we have 3 years for the plants to grow, produce lots of flowers, and now we're trying to get seed to seed within 1 year, because there's no point in introducing genomic selection in Miscanthus if you can't make seed quick enough to keep up with the genomic selection technology. And that in itself is a big challenge. So this is the project in a nutshell. A sort of slightly complex nutshell. So here we have the training set population. What we've done is we've gone back to the parents of that population. So we have 2 populations, because Miscanthus giganteus, which is the main commercial clone grown at the moment, is a sterile triploid, and we're very interested in making more of those interspecific hybrids, which have this really great traits for biomass. So we're looking at the Sapriflorus and Sinensis separately so that we can understand how they both work. So we've recreated effectively the siblings for the training set, and then we're using these models to go through 2 generations within this 3-year project. And just to reiterate, usually each one of these generations would take 3 years alone, so we really need that speed breeding component to make this work. So our training set was the most up-to-date version that we had in the breeding programme at the time. We were using Germany because Aberystwyth is not the most luxurious place for Miscanthus to grow, and it grows faster in other places. So we looked at these within species recurrent selection populations that we've had established, and they were genotyped in year 2 in Germany. So they were phenotyped in September 22 and March 23. We developed a new marker set for those, and the trial was actually dug up and replanted in Aberystwyth for validation. This is really important. And what's critical here is it's not the absolute values, but the rank order of performance. So we want to see whether that holds up across those different environments. So we took a range of phenotypes from both of those populations, different ones for the different species because different things are important. But for the purpose of this exercise, we're concentrating on the dry weight. However, we are looking at all of these in the validation set as well. These are all important components for other aspects of the biology and the breeding. So, key outcomes in terms of the genomic selection models. We have demonstrated for the first time in Miscanthus that we can do this. So we have the genomic, we've generated a novel markers system. We have done some analysis to get the cost of that down as low as we can. That is an available resource for the community now. And we've implemented those genomic selection models in both species. So we know that we can do this, and that means that we've got a real game changer for the breeding programme. And again, we've re-established that training set, and the phenotyping is, the first part is happening now, because it's year 2 in Aberystwyth now, and the final part will be in the spring. So then we'll have the validation of that different genotype effect. So that's one part. And then moving on to the speed breeding, we need to determine the environmental conditions that are required to accelerate plant development from 3 years to 1, to establish how we can get the flowering time under control and really get that seed set up as well. So Aberystwyth is quite far north for Miscanthus, so we have to send it south. So we've sent it to a little one of these Canary Islands off Africa here. So Miscanthus has a very nice time. It travels quite a lot within this project. So the plants are sent to El Hierro and planted in crossing blocks. The seed are collected from those plants and returned to Aberystwyth, where we germinate the plants. We perform the marker analysis on those seedling populations, and we generate those genomic prediction rankings. We select the top few of those, and they're sent back to El Hierro again, and the cycle starts again. So this is how we can achieve it here and now, but obviously we want to cut out all the travel, because what we want to do is be able to do that all in the UK. So, in order to do that, we still need to understand how to do these things, but now we're going to modify the light quality, the photoperiod, the fertiliser, the temperature and humidity in order to try and recreate the conditions that work in El Hierro, ideally at least as well in Aberystwyth. And so we've got some elements of the light recipe. In fact, we have an exciting tent that you can play in next door to pretend you're doing this. It's not quite the same as the expensive setup we have at home. And we've established some customised glasshouses. Paul Robson, who's here, will be able to tell you much more about this. There's a whole lot of innovations gone into generating these things. Lots of setup. I'm not going to go into those in detail. That's my time doing that. But the take-home message is really important. That in year 1, without any improved setup, we really had no flowering in Sacroflorus, a few tens of seeds in Sinensis. With improvements within the first sort of year of improvements, actually we achieved incredible seed set in Miscanthus sinensis in Aberystwyth, and in fact the seed quality was really high, so that was fantastic. We still had issues with Sacroflorus in year 2, so we had a lot of vegetative growth, but not so much of the flowering. And then in year 3, in the Sacroflorus, they've added in far red lights, and now we have flowering and obviously seed sets just happening. So hopefully within the next few weeks we'll know whether we have good seed set, and then we need to germinate those and see how well that's worked. But ultimately that's been incredibly successful. And additionally, in year 3 in Sinensis, given that we've worked in year 2, we are now looking at using temperature to actually modify the flowering time, and that's been successful too. So this was a really good news story. So key outcomes, we've managed to recreate the seed, we've gone seed to seed in a year, both on El Hierro, and now we've managed to bring that home, and we've managed to do that in Aberystwyth. Huge amount of work, project team, thank you very much to everybody, and I will say thank you very much.